have nothing to declare. Hi, my name is Stephanie Tremblay from Concordia University. I'm going to talk about a model for multivariate quantification that accounts for covariance between parameters. This allows to quantify deviations from a reference in white matter microstructure that can be applied in health and in disease. Multivariate approaches have recently gained in popularity in the neuroimaging field, especially in quantitative microstructural mapping, a field that has seen an exponential growth in the number of available modeling approaches in the past decade. Multivariate frameworks can potentially leverage the strengths of different modeling approaches and counteract the issues arising from the fact that imaging metrics are physiologically unspecific. It may also allow to capture the complexity and heterogeneity of biological processes that underlie behavior in a more holistic manner. These approaches appear promising in studying pathological changes in white matter in a number of conditions. However, the relationship between multivariate white matter microstructure and behavior is poorly understood in the healthy brain, which is essential if we are to then use these approaches to study pathologies. Moreover, the shared covariance between metrics is often ignored in multivariate studies. To overcome this, we propose a method that allows the quantification of voxel-wise multivariate differences in white matter microstructure. This method accounts for covariance between metrics and yields a spatially specific measure of the extent by which an individual's brain differs from a reference. We will also suggest a few example applications of our approach. For instance, I will show that the multivariate measure obtained can then be correlated to scores on various tasks and that it is behaviorally relevant. 10 voxel-wise maps were created from data of the Human Connectome project. This results in a feature matrix of 10 features at each voxel and for each subject. To compute the multivariate distance, we first need to set our reference group, the group from which the distance will be calculated. Here, since all subjects were healthy young adults, we set our reference group as the whole sample. So I computed group average maps and the covariance matrix between features on the full sample. We are thus computing the multivariate distance between every participant's brain and the group average of the whole sample. We chose to use the Mahalanobis distance, or MADI, which is the multivariate distance between a point and a distribution. With this method, covariance between features is accounted for by dividing by the covariance matrix, and this also scales the variables and transforms the data into uncorrelated data. Here we can see the correlation matrix between all features, where the high amount of correlation highlights the importance of accounting for covariance in multivariate approaches. Here we can see example distance maps in two subjects and the group mean distance map. We can see distinct patterns in different subjects, and the mean map shows areas where there is greatest inter-individual variability in our sample. To investigate the link between our multivariate measure and behavior, I then performed voxel-wise regression analysis between MADI and scores on cognitive and motor tasks. There were specific spatial distributions in the relationships of MADI with behavior, which suggests that our approach leads to a multivariate measure that is behaviorally relevant. We can then zoom in on some interesting correlations, like the negative correlations between MADI and scores on the dexterity task, and then extract the loadings or weights of each feature on the distance. Getting the metrics that contributed most to the multivariate distance in specific regions can help reveal potential underlying biological mechanisms. To conclude, I've introduced a multivariate approach that allows for the combination of multiple parameters while accounting for covariance between metrics. This gives us a voxel-wise specific measure of deviation from a reference. I also showed that this measure relates to behavior and that potential physiological underpinnings can be inferred by extracting metrics that contribute most to the distance in regions of interest. Multivariate approaches have wide clinical applications. They can, for instance, contribute to improving diagnosis accuracy in heterogeneous disorders, aid in identifying disease phenotypes, and in designing personalized intervention strategies. For an example implementation in a preclinical Alzheimer's disease population, I invite you to take a look at my digital poster. Thank you for your attention, and thank you to all the foundations that supported this work.